Peter Calabrese. I'm a professor at the University of Southern California, and the title of my talk is New Quantitative Biology Major at the University of Southern California. So 4 years ago, the University of Southern California introduced a new quantitative biology major. This combines biology and data science, and I don't think I need to convince this audience of the importance of this combination. This major was co-founded by Remo Rose and Mike Wooderman, two other professors at the University of Southern California, and I want to give credit to them right at the beginning. The next several slides list the course requirements for the major, and I apologize, these slides are a bit busy, but if you're interested, I also have a poster presentation at this meeting where you can study these requirements. We can see that there are a number of requirements in biology, chemistry, computer science, math, and physics. There are also a number of specialization courses, so the students do not have to take all of the courses on this slide, but 5 courses from this slide. One upper division elective from this slide, and several electives from this set of courses. I did want to pay particular attention to several of the courses. So the first is Qbio 105. This is a required Introduction to Quantitative Biology seminar. The students generally take this course in either their first or second year. And every week we have a different guest speaker who discusses their research. As you've noticed, many of the courses are in other departments. So the Qbio major is managed by the Quantitative and Computational Biology section of the biology department. And we teach these capstone courses. So all the students have to take at least one of these courses. So computational genome analysis, structural bioinformatics, or systems biology. And in addition, all of the students are required to enroll in at least 6 units of directed research in a lab that's approved by the Qbio committee. So USC is a large research university, so there's many groups to choose from, both at the undergraduate campus and at the medical school campus. So the students work on a wide variety of research projects, and there's a lot of exciting stuff going on. So the first cohort of students graduated this spring. There were 8 of them, and the major is growing. So 12 are expected to graduate the next year, 14 the year after that, and 21 the year after that. And most students are interested both in biology and math and computer science. So if this major didn't exist, many would either major in biology or a math or computer science or engineering major, or perhaps try to do a double major. After graduation, roughly 1/3 are interested in medical school, 1/3 in graduate school in one of the sciences, and 1/3 at working in a biomedical company. Also, a few are interested in law school. So again, this year, the first group graduated. Of the 8, 5 are going to graduate school in science. 1 is starting an MD, PhD program, and 2 are employed and applying to medical school for next year. So one of the biggest challenges we experienced is the university was very worried about students dropping the major and needing an additional year to graduate in another major. So this major is a challenging one, and it's drawing on many different skills. So currently, USC students have to apply to the Qbio major, and we've really been attracting some excellent students. And since we're a relatively small major, we closely monitor the students' progress. And so far, at least, only a very small number have left the major, and it's usually because they don't like the computer science courses. I also wanted to discuss Qbio 401. This is the Introduction of Computational Analysis of Biological Data course. This is not required, it's one of the electives, but I teach it and I developed the course and I really enjoy teaching it. So students get their hands dirty analyzing genomic data sets. We have weekly computing assignments in either Python or R. So some recent weekly assignments have been to find all the open reading frames in the coronavirus sequence, to graph Illumina quality scores as a function of read position and number, analyze multiple alignment files from the UCSC genome browser, and to compute bootstrap confidence interval of average pairwise diversity for different populations in the 1000 Genomes project. We also did in spring 2019 another project where the students collected water samples in the Los Angeles area, for example, the LA River, before and after rain, and tap water and other samples. And then we had a budget to do shotgun sequencing to sequence all the bacteria in the samples and then analyze the results. So this spring, we were planning to do a similar metagenomics experiment on different soil samples, but unfortunately, coronavirus stopped our plans. So again, my name is Peter Calabrese, and here is my email, and I'm happy to discuss any of this with anyone if you're interested, either the major or the Qbio 401 course, and thank you for your attention.